The purpose of this video is to discuss the recent understandings related to iron metabolism in dementia. This video will begin with an overview of what dementia is and how it works as an umbrella term for other neurodegenerative diseases. Following this, we will discuss how iron works in the body and what iron metabolism is. And finally, a discussion on recent research and understanding on iron metabolism and dementia, which will include topics such as the Fenton reaction and iron accumulation. Dementia is an umbrella term used to define symptoms associated with impairment in cortical function and can be categorized by progressive decline in two or more cognitive domains. These include memory, learning, language, performance of everyday activities, and the ability to communicate. These symptoms in dementia patients is caused by neurodegeneration. This involves synaptic failure and inflammation due to damage or degeneration of the cerebral cortex, most typically the hippocampus, which is responsible for memory. However, as degeneration progresses through the brain, different aspects of everyday living can be. These include the left hemisphere, responsible for language. Here, the patient may struggle with finding the right words to say. As we progress to the temporal lobe, which is responsible for the visual system, here it may become difficult for the patient to recognize faces. Additionally, damage to the frontal lobe can impact decision-making, planning, and organizing. And finally, if damage occurs to the parietal lobe, the patient may experience difficulty judging distances in 3 dimensions. The majority of dementia patients are diagnosed after the age of 65. However, diagnosis of dementia before the age of 65 is also possible and is defined as early onset or working class dementia. The term dementia can be subcategorized into many different neurodegenerative diseases. These include Alzheimer's disease, frontotemporal dementia, dementia with Lewy bodies, and vascular dementia. Although different diseases, they share common symptoms such as degeneration of cells within the brain that lead to memory loss and eventually difficulty in performing independent everyday tasks. Alzheimer's disease is the most common type of dementia worldwide. It accounts for approximately 80% of dementia diagnosis. This disease has symptoms such as memory loss of language, and confusion. This type of dementia can be categorized by B-amyloid plaque deposition and neurofibrillary tangles of hyperphosphorylated tau. In simpler terms, B-amyloid plaques are a form of protein formed by the breakdown of larger proteins called amyloid precursor proteins. In a dementia patient, B-amyloid proteins are found at an abnormal level within the neuron and clump together to form plaques. These then collect between cells and disrupt cell function. Additionally, neurofibrillary tangles are also a protein called tau. These abnormally accumulate within the neuron. Instead of binding and stabilizing microtubules in Alzheimer's, these tau molecules bind to each other to form tangles within the neuron, which then harm synaptic communication between them. Iron is a very important mineral within the body. It aids in many enzymatic reactions such as the synthesis of DNA, RNA, and proteins, as well as aids in the synthesis of myelin that is vital for brain function. Additionally, iron is a mineral that's main role in the body is to help make hemoglobin and myoglobin for red blood cells to help oxygen transport. Iron metabolism is defined as the breaking down of chemicals and nutrients within a cell. In the body, iron metabolism is tightly regulated to maintain homeostasis. However, once homeostasis is altered in the cell, this can influence iron accumulation and therefore allow B-amyloid plaques and neurofibrillary tangles to form, leading to cell and tissue damage in the brain. As we now know, dementia is often related to increased age. However, with increased age also comes increased iron deposits within the brain. Iron accumulation, specifically unshielded iron, can have negative effects on the homeostasis of the cell and can then lead to impairment of normal cognitive function and behavior through something called the Fenton reaction. Research has shown that when iron is unshielded, meaning it is able to be reduced by cellular reductants, in this case with molecular oxygen, it becomes cytotoxic and becomes able to generate hydroxyl radicals and hydroxyl anions within the Fenton reaction. This reaction occurs when there is an imbalance of iron and a catalytic process occurs where hydrogen peroxide is converted to highly toxic hydroxyl radicals. These radicals then go on to damage cell lipids, protein, and DNA structure, and function and can ultimately lead to cell death. The accumulation of iron due to poor iron metabolism in the brain precedes aggregation of the protein tau and formation of neurofibrillary tangles, which consequently cause protein and DNA oxidation, lipid preoccupation, and accumulation of advanced glycation and end products in those who have Alzheimer's, and thus influences damage to cells and brain tissue. Research presented by Anderson and Fraser in 2017 stated that excess iron may catalyze reactions that produce reactive oxygen species and consequently cause oxidative damage to cells and tissues. Furthermore, another research study that was conducted by Yuan et al. In 2011 presented that results from post-mortem brains showed increased iron accumulation in those affected by Alzheimer's disease. As seen in the image, the non-dementia affected brain is larger and has more tissue with less room between sulcus. However, when looking at the dementia affected brain, it is visibly smaller with more room between sulcus. This is due to the Penton reaction causing cell damage and tissue damage in the Alzheimer's brain. Despite an increase in research into iron metabolism and its effect on neurodegenerative diseases, there is still much more to discover on the topic and further research is needed. Thank you for listening to my presentation.